creating transgene expression cassettes to activate the molecular mechanisms that control neurotransmitter release. So within the brain, you have the nerve cells, which are the basic building blocks. Action potential is sent from the dendrites to the axon terminal, in the form of neurotransmitters. These neurotransmitters bind with synaptic vesicles ends and release those neurotransmitters to the receptors of the next dendrites. These neurotransmitters within them contain a protein called synaptagmin, which we're trying to determine the function. We're going to determine the function of the synaptagmin protein by creating transgene expression cassettes. These cassettes can be constructed in the form of this, with neurotransmitter new specific promoters, which regulate gene expression, synaptagmin itself, the protein that we're trying to find the function of, the MVI intron, which does not code for a segment of DNA, but instead is used to interrupt the sequence of genes, the polyadenylation signal, which is the synthesis of messenger RNA, and our EGFP protein, which is used to determine whether or not we've successfully created a transgene expression cassette, and it's visible under a microscope. So next we're going to run enzymatic digest of the inserts, and we're going to be taking them out of the insert and placing them into the vector. Placing the vector, they're going to be light at 16 degrees Celsius, which is not too hot where the cells don't want to recombine, but not too cold where no reactions taking place. Next, we're going to be performing calcium chloride transformation on the E. coli cells, in which we chill them on ice and then run them at a 40 degrees Celsius water bath, in which they become chemically competent, which the pores open up so that the plasmid DNA can be absorbed. Next, we're going to be playing them on LB and ampicillin plate. The ampicillin will kill off any other bacterium that is not ours that we want, that already has a resistance gene, as shown here through the trend. Next, we're going to connect a PCR screen, in which the strands of DNA are denatured and then annealed with new primers and extended so that the cycle repeats and we have a multitude of copies. The mini preps are used to purify the DNA from its bacteria cells, as shown here. After that, we're going to perform gel electrophoresis, in which the mini prep gel is digested by the electrodes running negative and positive, which determines the molecular weight bands of the DNA. So the results are shown here. As you can see, this is the standard for comparison. Since the DNA is negatively charged, it moves from the negative end to the positive end, and determining how far it went is by molecular weight. From there, we're able to draw a conclusion that the expected band size were positively intensified by aqueous gel electrophoresis, which indicated that the ligation transformation was successful in creating a transgene expression cassette. Being that we're successful in creating the cassette, we're able to use that to be cloned to plasmid HDAd, which can be used for route production. This enables us to study the function of synaptagmin at the presynaptic terminal and determine its function in neurotransmitter release.